Hello friends, today we will be studying histology of connective tissue. Tissue. It is a group of cells organized in a specific manner to perform a specific function. It consists of cells and extracellular components. There are four basic tissues: epithelium, connective tissue, muscle tissue and nerve tissue. Connective tissue supports other three basic tissues of the body. It consists of cells, fibers and extracellular matrix. Functions of connective tissue are: it gives definite shape to an organ or body, supports other tissues and performs other functions. We can classify connective tissue into three major groups: embryonic connective tissue, present only during embryonic life, examples being mesenchymal and mucoid connective tissue; general connective tissue, which contains loose and dense connective tissue; and specialized connective tissue, which has specialized extracellular matrix, example being cartilage, bone and blood. This shows us the classification of connective tissue, where we classify connective tissue into general connective tissue and specialized connective tissue, and general further classified as embryonic connective tissue and adult connective tissue. Embryonic connective tissue contains the mesenchymal connective tissue and the mucoid connective tissue, while the adult connective tissue can be classified as loose connective tissue and dense connective tissue, and the dense connective tissue further subclassified as dense regular connective tissue and dense irregular connective tissue. What are the components of the connective tissue? As we said, it contains cells, fibers and intercellular ground substance. Cells are resident cells and wandering cells. Resident cells are fibroblasts, adipocytes, macrophages, mast cells, pigment cells and adult stem cells, while the wandering cells are the lymphocytes, plasma cells, neutrophils, eosinophils, basophils and monocytes. The fibers are of three types: collagen fibers, reticular fibers and elastic fibers, while the intercellular ground substance is made up of proteoglycans, glycoproteins and glycosaminoglycans. We now see the various cells of the connective tissue. Fibroblast. The shape is elongated fusiform. The structure, nucleus, is darker stained, large, granular, ovoid and containing well-defined nucleolus. The cytoplasm is pale staining, has branched cytoplasmic processes and is difficult to distinguish from collagen when stained with hematoxylin and eosine. And this diagram here shows us the structure of the fibroblast. Fibrocyte. Shape is elongated fusiform. Structure, inactive mesenchymal cell having a flat elongated nucleus with minimal cytoplasm, limited amount of rough endoplasmic reticulum, and this is how a fibrocyte looks like. Adipocyte. Shape, large, spherical, size up to 120 micrometer in diameter. Structure, these cells continuously store fat in the form of single fat droplet which enlarges and pushes the nucleus and the cytoplasm to the periphery, and that is what gives the signet ring like appearance to the cell, and that is what is seen here in this diagram, the signet ring appearance of the adipocyte. Macrophage. These are of two types, fixed and wandering. Fixed macrophages have irregular shape, short branching cytoplasmic processes and a kidney-shaped nucleus. They contain numerous lysosomal vesicles for digestion of the phagocytosed material. And this is how a macrophage looks like. Mast cell. Shape is ovoid. The size is anything between 20 to 30 micrometer in diameter. Structure, the nucleus is centrally placed and spherical in shape. Presence of numerous metachromatic granules in the cytoplasm and these membrane-bound granules are of 0.2 to 0.8 micrometer and contain heparin. Cytoplasm contains several mitochondria, a sparse number of rough endoplasmic reticulum, and a small Golgi complex. Lymphocytes. The shape, rounded cells, pleomorphic during migration. The size is between 8 to 10 micrometer in diameter, and on the basis of the size, the lymphocytes can be further classified into small, medium, and large. The small lymphocytes measure about 8 to 10 micrometer, medium measure about 12 to 15 micrometer, and large measure about 15 to 18 micrometer. The nucleus is rounded. Lymphocytes. Shape, rounded cells, pleomorphic during migration. The size is between 8 to 18 micrometers. On the basis of size, the lymphocytes can be of three types: small, medium, and large. Small measuring 8 to 10 micrometer, medium measuring 12 to 15 micrometer, and large measuring 15 to 18 micrometer in diameter. The nucleus is rounded, dense, rich in heterochromatin, and is eccentrically located. Cytoplasm stains a light blue and contains a few azurophilic granules in peroxidase reaction, few mitochondria, a small Golgi apparatus, few profiles of rough endoplasmic reticulum, and small number of lysosomes. Plasma cell. Shape, large and ovoid. The size is 20 micrometer in diameter. Structure, the nucleus is placed eccentrically. Cytoplasm is intensely basophilic because of a well-developed rough endoplasmic reticulum with closely packed cisternae. There are a few mitochondria scattered between the rough endoplasmic reticulum. The spherical nucleus contains heterochromatin radiating out from the center, giving a clock face, spoked or cartwheel type of appearance, and the same is seen here in this diagram. Neutrophil. Shape round, size 9 to 12 micrometer in diameter. The nucleus is multi-lobed. The lobes are connected to each other by slender chromatin threads which increase in number with age of the cell. In females, the nucleus presents a characteristic small appendage, the drumstick, which contains the condensed inactive second X chromosome. There are three types of granules which are present: small specific granules, 0.1 micrometer in diameter, large azurophilic granules, 0.5 micrometer in diameter, and tertiary granules. Eosinophil. Shape, rounded cells, pleomorphic during migration. Size is between 10 to 14 micrometer. Structure, sausage-shaped bilobe nucleus in which the two lobes are connected by a thin chromatin strand and surrounding nuclear envelope. Golgi apparatus, a limited amount of rough endoplasmic reticulum, and only a few mitochondria, usually in the vicinity of the centrioles. Eosinophils possess specific granules and azurophilic granules. Specific granules are oblong, 1 to 1.5 micrometer in length and less than 1 micrometer in width. Non-specific azurophilic granules are lysosomes, 0.5 micrometer in diameter. Basophil. Shape, rounded cells, pleomorphic during migration. Size 8 to 10 micrometer. S-shaped nucleus, large specific granules, small Golgi apparatus, a few mitochondria, extensive rough endoplasmic reticulum, and occasional glycogen deposits. Basophils have several phase receptors on their plasma lemma, approximately 0.5 micrometer in diameter, and frequently press against the periphery of the cell, creating the characteristic ruffled perimeter as seen under light microscope. Monocyte. Monocyte is the largest of the circulating blood cells. Size is 12 to 15 micrometer in diameter. Structure, it has a large eccentric kidney-shaped nucleus that frequently has a moth-eaten soap bubble appearance. The cytoplasm contains deposits of glycogen granules, a few profiles of rough endoplasmic reticulum, some mitochondria, free ribosomes, and numerous lysosomes. The periphery of the cell displays microtubules, microfilaments, pinocytotic vesicles, and filopodia. Golgi apparatus is usually seen near the indentation of the kidney-shaped nucleus. Now let us see the three different types of fibers. Collagen fibers. These are called so because they are made up of a protein called collagen. Collagen is made up of molecules of tropocollagen. Within a fiber, the molecules of tropocollagen are arranged in a regular overlapping pattern which is responsible for producing the cross-striated appearance of the fibers. And this is how the collagen fibers appear, and in between them we see the fibroblast cell. Composition. The collagen fibers are composed of a protein called collagen, which constitutes 30% of the dry body weight. Characteristics. They occur singly or in bundles. Examples, tendon and aponeurosis. The collagen fibers do not branch. They run in a wavy course and are white in color when fresh. The structure of collagen, each collagen fiber consists of small parallel fibrils. Synthesis of collagen. Collagen is not only synthesized by fibroblast but also by other cells, namely the chondroblast in cartilage, osteoblast in bone, osteoblast in bone, smooth muscle in blood vessels, odontoblast in tooth. Types of collagen. There are 6 major types. Type 1 seen in connective tissue proper, bone, dentine and cementum. Type 2 seen in hyaline and elastic cartilage. Type 3 seen in the reticular fibers. Type 4 seen in the lamina densa of basal lamina. Type 5 seen in placenta associated with type 1 collagen. And type 6 Type 4 seen in lamina densa of basal lamina, type 5 in placenta associated with type 1 collagen, and type 6 attaching basal lamina to the lamina reticularis. We then go on to see the elastic fibers. Composition of elastic fiber. These fibers are composed of protein called elastin. Synthesis of elastic fiber. Elastin is synthesized by fibroblasts and smooth muscle cells in the blood vessels. Structure of elastic fiber. The fibers occur singly or in bundles, branch and anastomose forming a network, can be stretched and are yellow in color when fresh. Examples of elastic fibers. These are found in ligamentum nuchae, large arteries and basement membrane. It provides supporting network in many situations like spleen, lymph node, bone marrow, liver and kidney. And this is how the elastic fibers appear. We then go on to see the reticular fibers. These are very thin, immature collagen fibers found to be continuous with collagen fibers, and they appear first during wound healing. Structure of reticular fibers. These are structurally similar to collagen fibers and are composed of collagen type 3. Examples of reticular fibers. They form supportive framework of lymphoid organs and glands and can be stained black with silver salts. Therefore, they are called as argyrophilic fibers, and this is how reticular fibers appear like. Thus, we have seen the general information about connective tissue, and also we saw how connective tissue is classified. We also studied the various cells which are seen in the connective tissue and the 3 types of fibers which are seen in the connective tissue. Next, in the next video, we will be seeing the specific slides related to the connective tissue. Thank you.